Hello? Hello. Is it working? Hello? Yes, it is. My thing's going up. G'day. It's not working, is it? I think, yes, it is. Good. Hello. I'm just going to wait a bit. Sunshine. How you going, sunshine rainbows? Today, I'm going to talk about nitrogen, a fair bit of nitrogen stuff. So for all those who want to know the ins and outs about nitrogen, what different types there are and how to use it, when to use it, I'll hopefully go through some slides and answer those questions for you. There wasn't much of a Q&A last week, so I'm not going to do much of a Q&A. Well, if you've got Q&A, it'd be good, but I'm going to show slides a bit this week again. But if you've got any questions, please save them, and I'll try and answer them later on. All right. Well, I'm going to start and go through this week's cool stuff that I found related to cannabis. Share screen. It's recording, yes. Allow. All right, I can't see what's written now, if there is any chat. This is really cool. Actually, I'm going to do that last. Hopefully someone will come in. Actually, hang on. I'm going to read. It's that one, isn't it? That's where I want to start, yes. So this is just some state by state for the people in the States on how your country rates, with the one being the most favorable for cannabis, and then it tapers off to 49 as the least favorable. So you can see wherever you live, like Idaho, yeah, if you're into cannabis, you might want to move states. Or Indiana, a couple of eyes, yeah, go to the green area. Not those areas, they must drink heaps. But anyway, that's the law. That's what's as of August 2021. Oh, it's not very current, but anyway. Next thing, this is pretty interesting. This is about auxin, and that's one of our plant hormones that we use, and is very beneficial for the plant. Auxin, like, it's critical. It's in regulating. This was a test to see how it works with its transcription factors down here, and it found out that it's the plant solidity and it's drought stress, and that's what auxin's very full on and very good for. As a chemical messenger, it influences gene expression, and then that comes down through these response factors, transcription factors, and then goes down through the DNA binding, and then with these, and then ARFs, the auxin response factors. But that proved that was really beneficial for plant salinity and for the drought stress. So if you've got high salts, if you mix your stuff up, and that's what high salinity is. Oh, this is pretty amazing. This is the difference from male and female effect on the endocannabinoid system, and amazingly, it's full on. In these different areas, there's different tests shown, excuse me, that the different areas affect men and women differently. Here, I'll just read something important. This infographic is based on experiments in rodents, found a few key differences in cannabis with response to males and females. In the liver, the males produce less of this chemical, less 11-hydroxy-THC than women. This means women are more likely to have more intense experiences with edibles. And with your sex hormones, while testosterone doesn't alter much from THC, there was a clear connection between estrogen, progesterone, and THC response, meaning that women's menstrual cycle can have a huge impact with THC, and it gets some relief off it. And for pain relief, down here with your noses, this was a mixed finding where men get better anti-nociception with cannabis for certain types of pain, but for other types in women who get the most pain relief. So yeah, it depends on the pain. Different types of pain you get, I suppose, works with different. In other words, your biological sex can have major impacts on the way THC, CBD, and other cannabinoids affect you. Yep, that's cool. What's the essay at the end? Have you ever noticed any of these effects? If you have, Oh, women should be edibles. Yeah, edibles hit women a bit harder. This test concluded by saying that. You tell me. And I like this. This is back in the day. This is I just can't believe this. In the 1930s, in American Medical Association, in their literature for general practitioners, it was taken out of their prescribing things of cannabis because of Henry friggin' Anslinger and about he loved his alcohol and shit. And you know, that's a bit of a history lesson. But yeah, and I just like the cool things down the bottom. This is what they call cannabin. That's some tincture from Cannabis indica, some extract, Indian cannabis. So it's from way back in the day, from the 1850s to the 1937 when it got outlawed. Well, I think it got outlawed a little bit before that, but anyway, that's what this thing says. Ploidy, yes, polyploids. They are extra chromosomes. That's what it means. Ploidy, yeah, it means extra chromosomes without breaking those words down. You get extra bonuses from it. I've been playing around with them for a couple of years now, and there's tetraploids, which has 4 sets, and triploids, which have 3 sets. This is just a thing. So this is some myths that people think. I thought this was pretty cool, so I cut and paste this little thing. So having these 3 sets of leaves, that's a gene mutation. And the same thing here down with, that's called horde phyllotaxy. Horde phyllotaxy, that's what that's called. When you get multiple branches, remember when it's you have the BRC branching gene expression dominant, and you'll get multiple branchings can come out. Your facetation, that's where, well, there you go, you can read as well. But that's not anything to do with polyploids. That's just a gene expression as well, because you know your plant's got 40-odd thousand genes, so it's going to throw dominance in different ways, and when they co-work together, they throw different expressions. When you mutinize things and hurt them, they'll also throw different traits. Anyway, with the Ploidy, visually indistinguishable. I wrote to this person. They're not visually indistinguishable. They've got the center leaf is very, very prominent in mature leaves. So you can't tell when they're small because, see, they look like this. But when they're mature, it just looks like someone's got a giant middle finger. And you can also tell through the stomata. So if you pull it under a really high-grade microscope, you can see the stomata and measure their lengths, and they're about double, because they say 20, about 15 micrometers. Micron? Oh, just had a smoke, sorry. Yeah, micro. They're about 15 odd to 20. I don't know. Yeah. Anyway, the polyploid ones are about 30 upwards, and they got lesser numbers too, lesser frequency. That's how you can tell visually. Because I'm playing around with this 4 different types. I'm playing around with the auto polyploids and the allo polyploids, and that just means their expression of chromosomes. Did I make them from 1 set, or did I make them from 2 sets of chromosomes? That's what that difference means. And I'll get out of this. It's people. Not many people are doing that, to be honest. There's only a few people I find that are actually doing it. This is rad. Hopefully there's a few people watching now. The environmental factors that are affecting deterioration of cannabis post-harvest. One of my courses at the moment is on post-harvest, and it goes unbelievably into detail. Like, there's so much maths that you can do and stuff, and it just goes into, anyway, the relative humidity has a direct effect on microbial growth in association with temperature. And I've gone on this a hell of a lot about the AW, or the water activity level. There it is over here, water activity level. When you do your maths, it's AW. I suppose explain this one first. This is pretty cool. So if you got your humidity, moisture content, it's this one. If you got your relative humidity, and if it's above 65% when you're drying it, so you've gone and chopped it down and you're hanging it up and it's drying there, put your meter in there, leave it in there. And if it's above 65%, this is what you can get. You will get fungi and insects can grow. It creates the right conditions for them to proliferate. And if it's above 90, that's when bacteria grow. But no one has that. Shit, you aren't dripping in there. So 65. So if I was doing big when I did my stuff in Canada, when I was legally doing it with the ACMPR over there, I set my dehumidifier at 260 for the first 2 days, and then I think 55 for the first 2 days, and then I put it back to 60, and that's how I dried it. And this is just a chart to prove why that is beneficial, because if you go below that, insects can grow, but microbes don't grow below 65, meaning fungi and bacteria. And if you go below 35% humidity, nothing grows, no insects, no nothing. So that's just, I don't know, it's just so important. And you can see the temperature is also relative down here too. So if you've got it down at 20C, which is 20C is 68 Fahrenheit, or up at 30C, which is 86 Fahrenheit. So you can see, look at the different growth here. So below 20 with a high humidity, you're going to sort of not get microbes. So if you've got a higher, you're stuffed if it's above 65, all right? Nothing you can do. But if it's a bit lower, you can play around with it, juggle these numbers, because temperature is relative to humidity. Over here is very cool. I've wondered this for a long time, the activity that goes on in cells after post-harvest. And this proves it. It shows that, lipids, all the fats and oils and stuff, once the humidity stays above 60%, they're still going to break down. Non-browning enzymes, they still are effective at about 50%. Hydrolytic reactions that are involved with water, so the cells have still got, if they've still got a high amount of water in them, which a plant has 70% water, and now on harvest you might get it down to, I like mine at about 9 or 10%. So they still have water in them. So if you've got 50% water, you're still going to get degradation. But look over here with the enzyme activity. It's got to be really, really high. So this is cool. So even above 60%, it's still going to do a little bit of breakdown. But right above 80 is when you get big time problems. And we've already gone through about the growth of the bacteria and the yeast above the 65% and the 90 for the bacteria. Combination. Temperature and humidity. That's what I was kind of talking about up here that went through it. So the post-harvest diseases are more severe when relative humidity is above 90. No joke. No one ever gets above that. But this is pretty cool to watch. This hasn't really been brought to my attention, but from playing around with it for years, I've worked these numbers out, and this is some charts that proves that. Something that's cool. This is amazing. This isn't really cannabis related, but I thought, well, I got to bring it up because it's just damn cool. Because it's to do with cancer, and that's kind of cannabis related. What is it? The figure summarizes the strength of current events related to the estimated number of patients. No, it's not. That's the wrong one I'm reading. Researchers at the State Michigan University have shown that locusts can not only smell the difference between cancer cells and healthy cells, but can also distinguish between cancer cell lines. So these are cool. So if you've got any cancer, maybe, I don't know, train them, work with them, because they're your friends and they're going to detect that stuff. How cool is that? This is electron scanning microscope of a University of BC, how they got in and they worked out the pathways for the trichomes. And I see a THCG, which is the cannabinoid family tree. That's where it all initiates. They just worked out more pathways for that to happen. And I thought it's another cool photo. They peered into a cryophys. This is a purple Kush cell. So that was something pretty snazzy. And this is the figure that summarizes the strength of current events related to the estimated number of patients using cannabis based on medicinal products for certain conditions. So on the bottom, the x-axis is relatively number of patients. So low or high have said that evidence, and up here is the actual evidence. Is it low or high? So this is from their tests, and this is from the person's reporting. So you can see over here, chronic pain, pretty much everyone says it works, and evidence says it pretty much works too. And up here, high, intractable epilepsy, local epilepsy, and I don't know what that means, but it works really good with it because it slows down the neurons, and that's what the problem with mine is. My neurons fire too fast. I'm always it's peaking this and that, and it slows it down and allows them to actually communicate properly and not misfire. But this is just a bit of, it helps everything. medical cannabis is the future. Government, speaking of government, oh no, this isn't the government one. This is a, what do I call it? Intercalary meristem culture. He called it, he said a different way of cloning. But the way you're cloning it, it's from intercalary sections. That's where the meristem is, underneath that. And you wring it out, and then you have the, just underneath the phloem there, and that's on the outside, is phloem parenchyma cells. And that's where they will proliferate, because the parenchyma cells are the ones where you tissue culture from, and they are undifferentiated, meaning that they can form anything. You can turn them into anything. So that's all you do is you wrap it up keep it wet, wrap it up, and it throws roots. And that's what he's done. So he's cut it off nice and neat, wrapped it up, kept it moist, and it's throwed roots. Then you cut it off. So this is a bonus for people on low plant counts like us in Australia, or in the ACT you can have 4. Soon you'll have some. Everybody will bloom and have some. So yeah, if you want to stay legal, which I really advise to work with the law, because otherwise they'll hurt you, you do this. So you have your plant and you think, all right, well, I want to get some cuts. So you do it on the plant, and then you're not increasing your plant count. So until that's cut there, this isn't another plant. So it's just something cool. Signs of infection. What's this? Oh yeah. Oh, this is really cool. This is I had an infection, and I thought, well, how do I find out what's going on here? So I took a cutting, I cultured it, and it told me what it was. So this is signs here, and it turned out it was dampening off from, what was the microbe? Pythium. And this is the signs of it. So you see how they're curling a lot, and they're doing the Tarko effect. So it's saying the closis. Looks like magnesium. And I thought, oh shit, I knew something was up anyway. And then I recovered it, and you can see this is its recovery. So see down here, look at the leaf margins on the end, how they're all tilted up. Look at this one, it's perfect. That is the stage where at the leaf it's recovered. So see, look, it's main mid-rib vein. It goes down here. So it's only this side of the leaf that actually got the infection, because see, these margins are fine. So just that section there, and it's recovered. So I thought it's pretty snazzy. I let the immune system kick in, and now this is giving it a systemic acquired, it's gone through the systemic acquired resistance. So now this, because nothing else got affected, so because they already had a systemic acquired, I got fungal resistance in its genetics, but this is a seed, so this didn't have it installed. So now it's been installed. So that's why I worked with it, and yeah. Oh, this is some bad infection signs. Look at that, it's really bad. And now it's fixed. Hey, no worries, mate. Oh, and this is some wow factors. That's just a wow. That's just wow. That's really cool. Look at this. You can see the size of the bag there. Very good job. Now, this goes mostly to people in the States. So when I was in Canada, you can do this over there, and I did that. So if you've got something this big, there's no chance you're going to dry it. You've got to really break that up shitloads. So you'll halve it again. You'll probably put that in about 20 different pieces, just so it can dry out, and you might even crumble it, and just to get the consistency of the drying, because otherwise it dries so fast on the outside and so little on the inside, and it changes color decently, so you'll know it when it dries. That's just a wow, though. That's wow. And another wow. This is so rad. Look, I'm going to zoom in. Imagine cruising home from work each day or something, and this is my driveway. You'd park up on the street and you'd bloody walk in, and you'd just be like, your tongue would be hanging out, and you'd be drooling and shit, and you'd find you'd be lying down halfway along, the sun would be down, you'd be sleeping here. Well, I don't know about you, but I love this. This is it's just really nice. Actually, I should pull that as a screenshot or something. I just like it. It just goes right into the distance, and your house is just around the corner. It's so what dreams are made of, eh? And this is this is some Pink Death Bubba, number 2. So this is a strain that I made from Island Pink and GML's Death Bubba cut. and it's just in different colors. They're in here, and I put it in because I've been outdone massively. Look at that. Look at that. That's not me. I wish it blimmin' was. I'd claim that so fast. They've gone and unlocked the anthocyanins. So you can see it's got the reds, the purples, and it does blues as well with anthocyanins. So decent flavonoids have been unlocked. I made a video on that. If you just want to do that too, it's the temperature you can do. I do it through temperature, and I've got a pretty good instructional video on how to do it. I made it a few years ago. But that's just so nice. And you can see they've got the carotenoids there too. And then there's the chlorophyll pigments. Do you know the chlorophyll are one of the most abundant, they are the most abundant molecule on the planet. And then it's carotenoids, actually the second, the yellow with the beta carotene in it. That's just pretty. Go back to my one. Look at my one. Look at this amateur. I'm still proud of that. I don't care what anyone says. It's not. But yeah, is there any more? Oh, that's it. No, I'm sure there was something else. Oh, that's right. There was a. Is that all there is for there? Oh, yeah. There's the cannabis on the website. So I'll show this. It's the grant for Australia. Good on them. So we've got finally stuff might happen in Australia. So a $5 million boost has been given in April for research at La Trobe University to do next generation crop protecting research training that will innovate medicinal cannabis production and enhance agriculture industries. Because remember, the first medical cannabis place went into liquidation a few weeks ago. So they're thinking, how can we stop this? We can't do it. Why isn't it working? Because they don't hire people like me and you who have experience with the actual plant. They get the people who, it should work like this, it should work like that. Well, it doesn't. But yeah, this is just props. Good on Australia. You know, at least they're trying. It's a start. But see it, it's just a start. Actually, that's all I want. I don't know if I want to show that one. That's a bit. Oh no, this is pretty good. I will show it. These are the 10 non-cannabis plants that contain cannabinoids. So if you're struggling to get cannabinoids in your area, find out if your legal regulations and see if you can get some other things like, well, in this list. And then you'll be able to turn on the receptors, the THC, the CBD1 and CBD2 receptor when you induce your THC and get those reliefs. Endo inside, phytoplant, synthetic man-made. Yeah, good. No. We're going to go down. Yeah, and CBN affects that receptors, binds with that too. Interesting, isn't it? That's the points from where it engages in the body. So that's where your receptors are. That's where it can turn it on. So that's why it's good for the females with their menstrual cramps. Look at that. Woo! But what was the point? That's right, the names of the things. So non-cannabinoids, non-cannabinoid plants that produce, oh yeah, that's what the whole summary was. The names, black pepper, cone flower, looks pretty cool. So they have really low amounts, but they will engage your receptors if you have them. Helichrysum contains high amounts of CBG. Black truffles, they contain a chemical similar to THC. Japanese liverwort, it will engage your Japanese liverwort, contains perotentinic acid, which binds to CB1 receptors. Kava, that's popular in Indonesia. Electric daisy, wow, it's pretty electric, isn't it? It engages the CB2 receptors and can regulate pain and inflammation. Make your own smokable blend. Oh, righto. Oh, yeah, there's a bit of a smokable blend for those people who are struggling to get cannabis because it's so damn expensive. And if you can get relief some other way by the same molecules, there you go. I wouldn't advise it, but it's an alternative for good health. I'll scroll up now and say good day and that and see if there's any questions. And then next I'm going to get onto is about nitrogen. And this is really cool. There's some really interesting things about nitrogen. So how you going, Sunshine Rainbows? G'day, Genetic Memory Farms. And Koga Ganakaru. how you going? Chris D, good vibes. Lene M, cheers. Oz Indica, hello. Donnie Marynuck, cheers, Grow fam. And Brett Chanter, that's a familiar name. Hello, Mr. Brett. Fantastic show. I have to go. I'm hoping you'll upload this later. I'll watch it then. Just for you, mate. It's there. And I also do it every week. All right, I'm going to share screen, get onto this nitrogen thing. This is really cool. So there's no questions. So think about it if you want any. Why is there a start one? I'll share again and get onto this nitrogen. I'm just going to have to find it. It was from my studies this week. There's some really cool things that can't get onto it quick. I'm trying to talk at the same time. Soil fertility and fertilizers. It's the name of the course. Oh no, that was phosphorus and potassium. This is the nitrogen. All right, get back to you guys and share screen. Thank you for bearing with me. Entire screen. Here you go. Ready for nitrogen fun. Now, I'm not going to have any questions, so I'll have to save it to the end. Nitrogen exists in many, many forms. Many bloody forms. You should see them all. It shows nitrogen release the amount. There you go. I hope you're reading too, because I don't really like reading much. Nitrogen, 78% of the atmospheric gas is nitrogen. It's called dinitrogen. And then it's converted to an ammonium and nitrite. Number 3 is nitrate. NO3 negative is nitrate. And they're the forms that the plant's available for. So it only takes it up in those 2 forms. So if you get nitrite, which is NO2, it can't accept it. It's just not available to be taken up to the form. Every other form of nitrogen has to be made into this form by microbes for the plant to suck it up and for it to be available. That's why microbes are so important. And all your synthetic fertilizers you get, they've all got this shit already in it, ammonium and nitrate. You can't have ammonium nitrate because that's what bad people use. Anyway, oh Jesus, don't talk. All right, basic process. I'm going to pretty much skim over this because there's a shitload of information here to absorb, and I want to get to the key points. The soil process, there's 3 things that nitrogen happens in the soil: mineralization, nitrification, and immobilization. Mineralization is where it turns into its minerals, that's actual mineral. Immobile is where it's immobile and like in an organic form or in like a DNA bond or a chlorophyll molecule, something like that. It's bound up somewhere and it's not made available. That's what it means by it's immobile. So it can be inorganic or organic. Organic just means it's attached to a carbon chain when it's made immobile. Nitrification is when it gets nitrified and when oxygen acceptors come in and it changes its state. And actually there's volatilization, here it is down the bottom. So crop removal, it can be from leaching, water, denitrification, volatilization where it changes it back into a gas, and erosion. So the way that you put it into the soil is through your biological nitrogen fixation, fertilization, or your organic additions where it can break down, and microbes will break and fungi will break that down. And your nitrogen fixation, actually it's going to go into a bit of that, I'll explain it all. This is the basic chart of it. I don't know how much detail you want me to go into. Actually, tell me now how much detail you want me to go into. And then we've got a 30-second delay, so I'm going to read the questions after now. I'm going to stop sharing it, and then I'm going to see if anyone's written anything. And if anyone, I'll just go off that because I don't know what you want. It's really, really informative though. It helped me. So I just would like it to help you too. I've got a really good understanding of it all now, so I could probably relay things really good to you. All right, next. I'm going to stop it here and see if there's anything written. Nothing written. All right, I'm just going to continue at a fast rate then. Share screen. Symbiotic and non-symbiotic. Symbiotic works with the roots, mainly legumes, and it's rhizobium. We want non-symbiotic in cannabis. That's the types of biological fixations by microbes we're looking for. Azos. I'm going to skim through all this stuff. Didn't say anything, so I'm sorry. I don't know. I'm just not going to go into it. It's pretty cool. If you really want it, you can get back to it. Synthetic or industrial processes. This is how to make it. This is its different effects. These are its different forms in the soil. So the inorganic forms, it's nitrogen, nitrite, nitrate. The bad ones, nitrous oxide, nitric oxide, elemental gas. Na3 are most important. With ammonium and nitrate are most important forms of plant, and they can only ones that can uptake. Here's something important. Usually it comprises of 2 to 5% of the soil. Nitrogen. The total amount of nitrogen available to a plant is lower than that. So the amount of nitrogen in a soil is only 2 to 5%. Because remember, a plant, it requires 1.5% of its dry matter is nitrogen. So that's all it requires, because 96% comes from 3 elements, your carbon, hydrogen, and oxygen. And then the next one under that is your nitrogen, which is 1.5%. So it's so little it requires, but it has to be available first. So that's why nitrogen deficiencies are so important, so prominent, is because they mostly in this form, the nitrate form, and that has a negative cation, a negative, which is a anion, which is the same as soil, so it doesn't bind to anything. So it can run off easy as, where ammonium will stick to it, so it's a lot better. Organic forms, and that's how it's bound up. It's in amino acids, sugars, DNA complexes, proteins. This is how mineralization happens. I've explained. Actually, I'm going to skim on this because has anyone written anything? I'm going to see if you want me to really explain stuff. No one. All right, I'm going to skim over it now. Mineralization, there are a few ways there. Mineralization and mobilization, it's just how it happens. I'm not going to go into it. And it depends on the CN, your carbon to nitrogen ratio, because the more carbon that you have in there, the slower it's going to be. The more nitrogen, which is your greener materials, the faster it's going to break down, because that encourages bacteria. Mineralization, nitrogen and mobilization, mineral happens with time, because after time it starts up here, nitrogen availability with the high CN in the soil, and then it gets used, and it has nothing. Then everything dies and goes green, lays down dormant, it becomes locked up again, immobilized, and then it starts getting broken down and becomes mineralized again and becomes available. That's the cycle. Mineralization of organic nitrate, different processes. Yeah, good. Nitrification. Yes, we already explained that. There's no one's bloody interested. What's the key factors? I'm trying to just skim this really fast. I'm going to end this soon. Soil nitrification. Yeah. Soil temperature. This is a major thing. Soil temperature is a major effect when you're playing with any of the elements. The more of the lower temperature doesn't allow the microbes to complete their cycle. So things can nothing happens. So this is just for nitrification. So as you go here, and it comes up to 72, and for us, the Australian folk, 72 is 22. So see how it comes up here? I keep my soil always at 24. That's when my probe's at. And if it gets too low, I'll set up a device, like put up a heating mattress pad or something like that. You know, you put under beds, you put that through a thermostat, put the probe in the soil. That's how I did it in Canada. And I always kept it at 24C, which is right here. So it's perfect for nitrification and all other microbes, because your soil, your magnesium, your sulfur, those things also have the same effect like this on this scale. Soil temperature, that's substrate temperature. That's why people remember with DWC and that sort of stuff, they check their substrates. They always keep it at a temperature. reductions in nitrogen. This is the reductions of temperature based, so it's just how much it reduces. Influences, yeah, good. Does anyone? I'll just stop this for a sec. See if anyone wants me to explain anything. G'day, Ned Kelly. Good info. Thanks. Yeah, I'll skim over it. There's its losses and its ways it can happen. There's stages of different cycles. Yes, good. You don't care. So it's already explained that. Denitrification. It's important through temperature. Yeah. It's emissions. You don't care. Ammonia volatilization. This is pretty important. Ammonia volatilization, where it can change when you get your losses, how it changes from NH4 back to NH3 and it turns into a gas. That's how it can lost from the soil. So for people that go and put their fertilizers on the top, namely also urea, which has a big amount in it, it'll sit on the top and it'll just go back to gas and you've lost it all. So you've really got to bury it if you're going to use different types of fertilizers. And then it can also leach out. So if you've got use the right type of nitrogen, if you want it fast reacting, you'll. Actually, I'm not going to. That's a big thing to explain, and I'm just going to skim over it because it's not. It's technical, and I don't want to bore people. People I think are already bored already because there's no real questions. That's the stages of eutrophication where the nitrogen, potassium, phosphorus goes back into the soil, the water bodies, and it forms the algae. Cuts off the oxygen, everything dies. It also can make spores happen, and it's given lung problems to people in New South Wales because of our algae blooms that we get here from our high temperatures in summer. It's mainly a summer thing. The factors with the degrees of influence in nitrogen loss. That's full on. There's a lot here. I'm not going to go even close to explaining any of them. This is how more nitrogen gets lost. This is more thing for the organic people. Because people, if you're not into organics, you're just going to bottle feed it, and who cares what the end result is? You just want your yield. But for people who are organics and care about the planet, like me, this is very, very important. And you want to put in the amount and have no output. That's called a usage efficiency. Well, actually, we're going to come up to that. Environmental problems associated with nitrogen fertilizer. Yeah, methane methylglobia. It's a nitrate problem. Gets into humans. That's what's the problem in humans. It's a disease you can get from the groundwater being too contaminated. You can get acid rain from it being in the atmosphere. Eutrophication, which is the green-blue algae, and underground leaching. Did you know that I think nearly all of the US groundwater is contaminated? There's another chart I showed a few lives ago because of all of the pesticides and runoffs and stuff. So if you live in the US, you have to usually use a reverse osmosis system or some sort of thing for your drinking water and for your plants. Plant uptake, yeah, good. Thereabouts, yeah, it's inputs and losses. This is, here's your nitrogen use efficiency. NUE, nitrogen use efficiency. So you want to make sure the inputs equal your outputs, and that's going to save the environment as well as the soil. And if you're growing over and over, you want to save the friggin' soil, and you want to make sure it's in its soil food web is really going very well and functioning immobilization to immobilization, and just keeps going and breaking stuff down, making it more mobile and regularly. The roots will come here, then you'll plant next to it, and this one here for a no-till, and then that one will get broken down, and then it becomes made mobile, like that chart showed before with the big sine wave effect. So this is something that's extremely important. You can get it from inputs from the nitrogen inputs from the atmosphere, from beneficial nitrogen fixation of azos, because we use non-symbiotics, fertilizers, which, you've got to use organic. I've got a list later. There's a list later on that shows you different fertilizers you can get, and they're the ones that are elemental fertilizers that they are. And I couldn't believe how some of the high numbers. I thought Cool Bloom was high with its 40s. There's some really high fertilizer you can buy just from your agricultural shop. The reason why we haven't been told this is because hydroponics people want us to buy their shit and spend top friggin' dollar buying their shit. You can buy a half kilo bag of ammonium monoammonium sulfate or ammonium sulfate or some cool ammonium nitrogen fertilizer that costs $10, and it'll do you maybe a year or a long time. And your nitrogen outputs, when you harvest it, it touches it, then it breaks down and goes back into the atmosphere through the gas losses. Runoff's another way too. If you put it too close to the soil on the top there, it'll just run off with water because it's very mobile, especially in the nitrate form. You want it in the ammonium form, and then it'll bind to the soil particles. But remember too, you've got a binding strength that I showed a few lives ago. And down the bottom, it's sodium, and then it goes ammonium, the next one up. So if you've got calcium or magnesium, that's going to take precedence over the binding strength of your ammonium. Just, I don't know, something to remember, all these things. My microbiology teacher said, it's so hard to take all this shit in. How do you do it? And she said that you just listen and absorb it. Don't try and think about it too much, because that's when you'll confuse stuff. Just absorb it and then go next. And then after you've heard it a few times, it starts to sink in. And she was right. That's how it's happened for me. Because I was so confused at the start, because all this stuff, like, what? Yeah. Calculating the nitrogen use efficiency. Well, yes. And there's a few different ways you can do it. Different efficiencies, ratios. Removal. Yep. And I don't like the maths, so I'm not even going to come close to explain this to you. But there's definitely a peak period where it comes where you can use too much and you're going to get no yield. Is that what this shows? Nitrogen uptake is high. Where's the fertilizer recovery? So there's none at the top. Nitrogen use efficiency is 14%. 26. See, this is a shit use efficiency. So they must have put too much. That's what that proved. They used too much because their output was too low, high with the recovery. It goes on about nitrogen efficiency, relationship, symptoms. Know about nitrogen symptoms. Here we go. This is a thing I want to show you about the different types of nitrogenous fertilizers that there are in its forms that it comes in. Remember, this Cool Bloom was in. It's about 30. It's like a 0, 40, 30 or 50, 30 or something. Look at this over here on the right. So these are examples. So that's its name over there. Then there's the examples, what it's mixed with. It's blend. So your nitrate's mixed with sodium or calcium, and it's and then these are its contents of nitrogen, percents of nitrogen here. So look, 33% ammonium. Oh, that's the bad one. Ammonium sulfate. That's what I use. 26%, and you put a pinch in, and it alters the ppm meter so much. It's just really, really cool. I do that because of the cation exchange, because ammonium's a positive cation and the water's negative, so that means they bind up already. So I try and unlock that already when I put it in water. I don't put it in straight away. I try and separate that bond by letting it dissolve a bit and letting it react with the hydrogen and the oxygen that's in the water to bind it up with the oxygen. Oh, is that right? Hydrogen, oxygen. Yeah, bind it up with the water. I'm getting a bit too technical. It's even confusing me. But look, the urea, look at that, 46%. It's like up near Cool Bloom. And then you look, 82. What? So you can go source your agricultural supplies, source half a pound or 150 grams or something of what would you source then? Well, ammonium sulfate, that's what I use. Ammonium sulfate nitrate. But you probably won't be able to get that one on the end. So ammonium sulfate is the one I use. Calcium ammonium, that's a really good one because that's very neutral when it breaks down. These ones are a smidgen acidic. So CAN is actually really good. So I'd advise that. Calcium ammonium nitrate. There you go. And look at those numbers, 25, 26, 28. That's just massive. Calcium, so it's 25. Ammonium, 26. Nitrate, 28. That's just incredible. Just thinking about that, I should be sourcing this one, but it's got a high amount of calcium, and I've already done my maths with my figures with my enhanced vermicastings that I use, so I try and use the amount that the plant wants, so this might throw the calcium out of whack. But anyway, I've gone on about that enough. This is flow chart where it adds to this to form that, like you can. So that's got your calcium carbonate with your ammonium and your nitrate. There you go, you get your can. General characteristics. Ammonium, ready soluble in water. Nitrates, residual, yes. Ammonium, alright, amide. Why use urea? Yeah, it's high, but, and look, it's suitable for foliars. Isn't it amazing? I've never done a foliar for a nitrogen application. I suggest people, can you try that? Because they suggest that it really works. And from all of my studies on this, from this uni, all their shit really works. And I haven't tried it yet, but yeah, that's something cool. Everyone reckons it doesn't, but I've done a lot of foliar stuff in the past, and it actually does work. It's whatever. Stages of enhanced nitrogen use efficiency. To enhance it. This is cool, the color chart. All right, let's get into the color chart. Yeah, neem coated. This is ways to slow it down. So you would coat them on the outside of your fertilizer. So if you go and you get your can from your supplier, you can go and powder it up or put it with a coating around it. So neem coated, I really suggest. Neem's great. Neem cake has a 4, what is it? It's a 4:2:2 ratio. It's just that alone. That's brilliant for all round. And it's got so many benefits. Sulfur and polymer coated as well. And they will break down and then they'll slowly release the nitrogen that's inside of it. So it just helps with nitrogen inhibition, slowing up the release of nitrogen. And the right placement was touched on before. So if you've got one that you want released slowly, put it deeper. And if they break down like slow, you can put it deeper. Ones that break down fast, you might put it closer to the surface because it's going to get used quicker. Without going into too much detail. Actually, what was that? That's about dissolving. Prilled urea takes little time to dissolve in water, where granular urea takes lots of time to dissolve. See, that's an example. So prilled, you would, if you're using that, it's going to take a lot of time to dissolve. So it's going to wash off easy and break down easy. A little time to dissolve. Yeah, so it's going to wash away and break down easily. So you put that in the right area, and the same with the granular. Where granular takes a long time because it's bigger. You can see how that's smaller. So that's just breaking it down. So you can judge that by the size of your fertilizers or your minerals that you give. If it's really, really tiny and powdered like nanoparticles, that means it might be actually in that mineral ion form, ionic form. If it's actually big and clumpy, that means it's going to take a lot to break down. On a case study about the placements, it just proved what we were just talking about. And this is pretty snazzy. Look at this, the nitrogen color chart. They use this in rice, but I kind of know it, but I don't know. If you can Google this image, it's called a leaf color chart. It'll come up with this picture, and download it, and you can make it up, print it out, put it in your room. And if you notice that, all right, shit, you want to be hovering about this one, these two, that's very high, and you're going into a chance in it being a toxic nitrogen. And under here, it's deficient in nitrogen. So it'll start to get chlorosis, because you'll see those, because remember that the chlorophyll is made up with magnesium in the center, and then it breaks off. It's magnesium cation, and then there's magnesium cation, and then the anions around it are nitrogen. So they start and break off, which gives you your deficiencies, symptoms. Another thing's chlorophyll meter, but it's expensive. Slow and control release fertilizers, yeah. We talked the nitrification inhibitors. That's a bit on the neem coating. All derived from seeds, and neem contains malacyanins, of which look at this, epinimbin. How easy is that to remember for Australians? Nimbin. It's got epinimbin in it. Diacetyl, salane, and azadirachtin are the active fractions which have dose-dependent nitrification inhibition action. And I use neem cake. It's bloody excellent. It's 4:2:2. So they've been soaked. You can see by the size of it, but how big of it to break down. It's sulfur coated. Different case studies from different soakings proved what we were talking about. This is pretty snazzy. This is the end. So get your questions ready, because I'm going to I think this is the end. So this shows you a roadmap of improving your nitrogen use efficiency. So develop and use cover crops to capture soluble nitrogen. So mulches retain nitrogen because they trap it from getting volatiles off and leached off from getting runoff. It'll stay in your mulches. Deploy no-till to reduce off-season mineralization. Improve soil physiochemical properties to retain that. Really, that means organic matter. Because if you have less organic matter, nothing's to bind to. The cation exchange ratio is very low, so it's not going to retain nutrients. Breed, that's something for me that I was going to do. Deploy soil microbes to promote mineralize in roots rhizosphere. So that means we've got to use our beneficials like the azos and what's the Benny's name? And there's we use non-symbiotic nitrogen fixing bacteria. Anything under that will work for us for cannabis. Improves and deploys symbiosis, because without microbes, minerals will stay in their form, which won't be broken down. They'll be immobilized. So we need them. Develop association nitrogen fixation. No, that's with them. Develop noduling. No, that's something that I'm going to do. I'm going to put noduling in cannabis plants too, because in legumes they have big nods on them. They're like round looking things, and they contain nitrogen fixing bacteria, and that pulls nitrogen out of the air and exchanges it with oxygen, and they output nitrogen for the plant. So that's something, I'm not going to go into my stuff. Future scope. Breeding technology for nitrogen fixing, low cost. There you go. That's it. All right. Any questions? Actually, I'll put this up. No. Well, that's right, the invite thing. I'll put the invite thing up before I look at the questions. Copy, just in case anyone wants to come up and say something. That's that there. All right, I'll scroll up a bit. Oh, yeah, it looks like there's been a bit of action. Not bored, listening intently. Oh, okay. Thank you. Mr. Sunshines. Oh, good on you. Jimu Sema, thank you for your support. All water is toxic to some degree now. New studies just come out. Do you know everything is poisonous? Even water. There's a runner in the Boston Marathon, and he was a doctor, and he died in the Boston Marathon because of water toxicity. He had too much water. It filtered out all of the electrolytes in his system, flushed them out, and he died. So even water is toxic. And same with our plants. If we use too much, everything's going to leach out. Do you reuse the rest of the plant after harvest? Well, in an organic situation, yes, I do. I put all the roots. They get broken back down in vermicastings because all of my beneficials that I've used, they get contained in the roots, and that's around the rhizosphere. So they will get broken down, and hopefully it'll contain in the vermicastings, and I won't have to keep buying beneficials type thing. So yes, I break it down. And the leaves, they also get broken down. They get thrown in the compost if it's too green, or they can get thrown in the vermicastings. If you want to increase, you have to make sure the CN ratio is right. You know, the 20 to 30 is to 1. Sunshine, is neem cake and neem meal the same? Good question. Well, neem cake is the remnants after processing of the azadirachtin. So if you've got heaps of the seed and you want to process the oil, and you'll process it, and that's what's resulting afterwards is the neem cake. I think neem meal is something similar, but it might have more of the properties in it of the azadirachtin. I can't answer that. That's about the best answer I can give you. Sorry, mate. I'd be Googling it to help to break that question down to answer it. There's me. AussyCC, great stream, bud. Went well with cooking dinner. Thanks, GMF, for your support, mate. I'm glad you listened to it while you're cooking dinner. Lene, thanks for the stream with all the great info. Thanks for your support, Lene. Without you guys, I wouldn't be doing this. So, yeah, thank you. GMF, here in the Midwest, we consume about a credit card worth of plastic a week just through drinking. There you go. Confirmed. Unreal. Yep, microplastics are everywhere. That's something that if you want to make money, there's a few things, plastics, there's microbes now that eat plastics. There's my microbiology's brought up a few names of the different types. You can Google it and find them. I'd be suggesting to try and buy some of them, culture and proliferate them, and the government will give you money hands down if they can see a little tiny experiment or something that you've done in that area, because that's going to help not only the government, but more so it's going to help the bloody future for everyone, because there's too much plastic, as we know. Donnie Merrinak, another solid show again. Thanks, Aussie. And Sunshine, yes, Professor Google. Well, it doesn't, Professor Google sometimes is shit, because it's only as good as the people who are writing it. And I've found a few technical things, very technical, that could be adjusted. But in general, that's why I go to Wikipedia, because usually that's fact-checked. But Google, well, here's an example. Tilray, put something up, it'll come up under Google. Tilray, there's scientists that work for Tilray claim that when they hang their plants upside down, all the resin drains into the buds, giving them a higher, stronger terp profile for the Tilray. What fuckwits. Unbloody believable. That's an example. So, well, that, just to clarify, that's mega wrong. That's so not how plant cells work and operate. So yeah, good old Professor Google, but it's a start, so you can try and go off. But I always try and end on Wikipedia, or I just Google it and put Wikipedia, or just search on Wikipedia, because usually I can nearly trust all them. That looks like that's the end. Any questions before we log out? Because oh wow, it's nearly been an hour. Jeez, tell me what you thought, guys. Was today all right? Because I don't know, I can go on about and slow to show the slides, but they interest me, and I don't know if they interest you guys. So I need to know, I'd like to know that. Like I could have told you about phosphorus and potassium, like that makes up 90%, 91 to 98% of the soil, but it's all unlocked. It's all locked up and immobilized. So again, you've got to use your phosphorus solubilizing bacteria or potassium releasing bacteria to make that available for your plant, because that comes, I'm crapping on a bit more again. Anyway, I'm going to go, I think, because I don't know what you guys want, and I don't want to just talk here to myself. So I've got a comment, I've got a community section on my YouTube. You're welcome to post stuff there. Any suggestions or anything you'd like to see, I'll try and help out with that. It's about the only way you're really going to get me these days. Yep. So try and do that. I don't do any other social media, really. I do Zoom, but that's about it. Yeah. Thanks, guys, for listening and watching. 8 years, I appreciate it. And for others who couldn't catch the stream, thank you for watching it after the fact. I Oh, yeah, Jeff, I haven't said g'day to you. Good on you, Jeff. It's all good information. I appreciate your support. Real good show from Jimu Sama. I'm just learning by osmosis. Osmosis, that's a good way to put it. Yes, I didn't know much about nitrogen before today, just the ratios. Oh, sweet. Well, I hope that helped. Remember, if there's any question isn't a dumb question, because it means that you want some clarification. So that's the way I treat it anyway. So I don't put any people down or anything like that, because if they don't understand, shoot a brick. Well, they don't understand, help them. Don't put them down. Give them the knowledge so they can do the stuff that we do and stuff. Yeah. So that's what I'm about. So don't feel free to ask anything, related to cannabis. It's all good info, sunshine rainbows. If I think of anything, I'll let you know. Good on you, mate. Thanks, Jeff. Lene, I think everyone's interested and appreciates your info. Yeah, I'm just not sure about if it should be slides or more so about things that they're interested in or that. I just don't know which direction, that's all. But thanks, Lene, for your suggestions. Donnie, sorry I was late. Plants needed water second week of flower. They're drinking like crazy. Looking forward to the replay. Yeah, that's good. Good sign. They're drinking. Sweet. You must have a nice low pressure for your exchanges to get the drinking and transpiration happening. Good work, Donnie. All right, I'm going to sign out. Thanks, everybody, for turning up. I'll see you again next week. Oh, there was a bricks video this week that got released. No more videos. I'm not making any more. Yeah, I'm doing study. I'm looking at about me. They've been a bit low on the viewing rate and stuff, so I put a hell of a lot of effort in. So maybe next semester when it ends, I might. But yeah, none for a while. Just this. I'll be here though next week. I'll put something together, and depends what you talk about. I might talk about some interesting things, research topics from cannabis through the week, some health stuff for it, or maybe some interesting slides if it's interesting enough that's related to cannabis, because my plant physiology class, that's got a lot of technical stuff and you wouldn't be interested. I'd bore you to death. But the soil stuff is highly related, and post-harvest productions, that's quite related too, because we all dry and cure our cannabis, and no one really talks about that in detail with maths and different figures and microbes and numbers. And that's what that class goes. It's a 12-week course just on that. So I'm finding it like crazy in depth. So it's something I can elaborate on too. I'll wait and hear for your questions though and see what you want, because it's all about you guys, not about me expressing me. So I'm putting this show on just to try and help others. So it's about what you guys. So good on you, Skids Bits, and everyone else. Thanks for tuning in. Yeah. Skids Bits, Donnie Marina, Lene M, Jeff Papalia, Sunshine Rainbows, Jimu Sama, Jeff Papala, Genetic Memory Farms, and I saw Ned Kelly, Brett Chanter in the room, Oz Indica, Chris D, Kodja Karu. Thanks, guys, and everyone else for after the fact. Cheers, happy breeding, happy growing to you, and good health to you as well. Bye-bye.